Good afternoon. Hi, everybody. Does anybody have any questions that we can talk about before we start? Yes, so the midterm is going to be over all the stuff that we've done since the beginning, so it'll be over the first four modules. Um, so you can go back and look at the you can go back and look at the first test and see which questions you missed and kind of use that to see what you need to study. And you can also kind of use that as a guideline for what kinds of questions you'll see on the midterm. So the midterm will be over modules 1, 2, 3, and 4. It's a little bit longer test. It's worth 50 points. Your short test was only worth 20. I have in the syllabus that it was supposed to have been worth 30. But when I made it, I had forgotten to do that. So your first test was only worth 20. And your second test will only be worth 22. And that way they can be, will only be worth 20 also. And that way they can be interchangeable. So for the folks that had issues or got kicked out, or if somebody does really horribly on one and does really well on the other, we can look and see if we want to substitute that higher score so that people have can use the higher of those two scores, so they'll be the same, and then the midterm and the final will both be 50. So we can do the same thing for that. We can substitute whatever the higher grade is, if that helps people with their grade. So that was my intention, was to have the tests be the same amount, the two short tests be the same, and the midterm and the final be the same. So the midterm will be worth 50 points. You may see some of the similar questions to the first test, or they might just be over the same kind of topics. I've kind of been taking questions mostly from the things that I hit on when I do the WIMBA archives. So there's a little bit more in the PowerPoint than what we've done in WIMBA, and I've kind of been taking the test mostly from what I focus on in the WIMBA presentation. So although anything's fair game, the stuff that we really have kind of studied in more detail would be a place to start. The stuff that the assignments apply to, or the stuff that the discussion boards apply to, or the stuff that I really kind of went over in the archives would be something to look at. So on this week's module, there's actually two PowerPoints posted. There's the normal PowerPoint that I have posted that I'll show some slides from today. And there's one that I think it's called Basic Genetics. It's a short PowerPoint. It's kind of a review of genetics. I put it together. I had some nursing students several years ago that had gone through anatomy and physiology, Bio 137 and 139, and needed just kind of a short refresher on genetics because they were studying genetically related diseases in their nursing classes. And so I put together that short PowerPoint to kind of help review some of the basic concepts and some of the things related to genetic diseases. So it's a good summary. It doesn't have quite as much detail as what we're going to go into, but it helps with some of the terminology. So after we go through some of these things, if you're still a little bit confused on some of the terms, that's got some good slides in it about the terms. And I'm actually going to show a couple of slides from it. I've included a couple of those slides in this PowerPoint that I thought were good at kind of summarizing a couple of the things that are usually kind of confusing. So that's a good one to kind of go through for some of the terms. Doesn't have, like I said, quite as much detail as we're looking at, but that's what that other PowerPoint is. And it's got that orange background. It's from like 2003. It's from several years ago. It's an older PowerPoint. So I didn't clean it up or anything. It's figures from a different textbook. I just put it up because I thought it might be helpful for people. If you think it's just one more thing to look at, then you don't have to mess with it. But if you think it's helpful, that's kind of why I put it up. So we're looking at chapters 8 and 9. These chapters kind of fit together. They're talking about growth and inheritance. Chapter 8 kind of deals more with cell division. And then chapter 9 deals with what we usually call Mendelian genetics, as opposed to things like molecular genetics and those kinds of things. But cell division talks about chromosomes, and Mendelian genetics talks about chromosomes. And cell division talks about replicating DNA, which we talked about in a previous chapter. So we're kind of building on some of our concepts that we've been going through, and I'm hoping to kind of tie some of the stuff that we've been talking about together. And we can even go back to, the first chapter, we had an organization figure. Atoms become molecules. Molecules become cells. One of the molecules we talked about was DNA. Then we talked about how DNA makes proteins in cells. Now we're talking about DNA again and what's happening as all the other events around the replication piece, and how DNA is involved in inheritance of those genes that make those proteins that happen in transcription and translation. So really, after we get through chapter 9, we can go back and tie together some of the things from chapter 6 and 7, chapter 8 and 9, chapter 3 and 4, chapter 2. And I think that's one of the reasons why this is a good spot to put a midterm, is it kind of forces you to go back and start to study some of the previous chapters. And once you have some of this information, you might find some things from those previous chapters kind of clicking a little bit. Or you'll be able to start to see some of the connections and how they fit together. That's my hope. And I'm going to try to point out some of those connections to try to help you make some of those connections as we go through. So chapter 8 is talking about cell division. It's talking about two main ways that cells divide. One of those processes is called mitosis, and one of those processes is called meiosis. And those are both mechanisms of looking at how the nucleus is dividing. Cells can only come from existing cells. Cells can only come from living cells. We know a lot about the components of a cell. We can put phospholipids in a test tube and make a cell membrane. We can put proteins and enzymes in a test tube and make proteins. We can put enzymes in a test tube and make DNA. But we can't put all those things in a tube and make a cell that functions. There's some force that is in living cells that is not in all those individual components, even when you mix them in a test tube. So we cannot make cells just by mixing stuff in a test tube. A cell can only come from a living cell. So cells reproduce by a living cell dividing in two. And each descending cell, each of those daughter cells, has to have everything in it that that original cell did. It has to have a full set of chromosomes. It has to have some cytoplasm. It has to have some organelles. And so that's what replication was. Replication was doubling the chromosomes in the DNA to get ready for the cell to divide. And so nuclear division has these mechanisms that help to divide the chromosomes into two new cells. And then separate from that, the cytoplasm will divide. And your book gives a really good picture of how cytoplasmic division takes place. So I want to review chromosomes just for a minute. Chromosomes are kind of these units of DNA, these long pieces of DNA. Your book gave you a really good picture in one of the chapters where it shows the DNA coil, and that coil wraps around some proteins and becomes a chromosome. I don't know that I'm going to be able to find it off the top of my head, which one I'm thinking about. Aha, figure 6.2 on page 103. So we talked a little bit about chromosomes in chapter 6, and we're talking about chromosomes again here because the chromosomes had to be replicated so that they could move into these two different cells. And so now that we're talking about cell division, we're talking about these chromosomes again. Human body cells have 23 pairs of chromosomes, or 46 chromosomes. And almost all of those pairs are homologous, which means the chromosomes are pretty much identical except for maybe what specific alleles they have. And we'll talk about alleles in just a second. But they're going to be the same length, they're going to be the same shape, they're going to have the same genes. And so 22 chromosome pairs are homologous. The sex chromosomes are the only ones that are not homologous. Mitosis maintains that chromosome number. Every chromosome is duplicated so that each new cell receives 23 pairs of chromosomes. If we started out with 46 chromosomes and we didn't replicate them, each daughter cell would get 23 chromosomes. And then each of those daughter cells would get 11 and a half chromosomes. And then each of those would get 11 and a half, not 12 and a half. And then each of those would get 5.25 chromosomes. And then each of those would get whatever half that is. So you see that after just a few cell divisions, at this point we're not even to the point where the zygote is implanting in the uterus, and we're running out of chromosomes pretty quick. So we have to duplicate the chromosomes or undergo replication in order for the cells to be identical after cell division. And we talked about that a little bit there in chapter 6 when we were talking about those mechanisms of replication. This is a figure from this chapter, from chapter 8, that's showing we start with just a pair of chromosomes and we duplicate the chromosomes in replication so that when that cell divides, each of these daughter cells looks exactly like the parent cell. And then this is a figure from a different textbook that's showing that same thing. It shows two chromosomes. It shows a chromosome that's been duplicated. The structure shows a chromosome. And then when a chromosome duplicates during that replication, makes an exact copy of itself. Those two copies are held together in a segment called the centromere, which is in the center. The two copies are sometimes called chromatids or even sister chromatids because they're exactly the same. We've made an exact same copy. And then we distribute each of those copies to the daughter cells, just like in the previous figure that we looked at. So all of these events can fit into something that we call the cell cycle. So we're going to talk a little bit about the cell cycle. Now, your book talks a lot about the phases. And it's easy to get kind of confused because they talk about the phases of the cell cycle, and then they talk about the phases of mitosis. And mitosis is a phase of the cell cycle. So using the figure in your textbook might help you kind of keep some of those separate. And so I'll go through and show you that. If we want to think about the phases of mitosis, then you can remember, if you can remember IPMAT, IPMAT, you can remember the phases of mitosis in order. And then where mitosis fits into the cell cycle. So here's prophase, metaphase, anaphase, telophase, and this whole thing is interphase. And they break interphase into G1, S, and G2. And they kind of call those stages. So we have stages of the phases. So that's why I'm trying to caution you to not get too confused about the terminology. And if you use these terms, then you don't have to worry about what are phases and what are stages and what they're talking about that. So this G1 is a good place to start. G1 also means gap 1, or the first gap phase. Here the cell is growing, and here the cell is making cytoplasm and other cell components. Then this S phase, where they show this number 2, S phase means synthesis. And what they're talking about here is DNA synthesis. That's where DNA replication takes place. Step 3 here is another gap or growth phase, G2, which is another phase of growth for the cell. And the cell is here, it's making proteins, getting ready for the cell to divide. From step 4, the rest of this piece is mitosis. Ran into my keyboard. The prophase, metaphase, anaphase, and telophase that we're going to learn about in just a minute as the phases of mitosis. So in step 1, we're making proteins. In step 2, we're making DNA. In step 3, we're making proteins. Step 4 begins mitosis. And so in all of these steps, we are dividing the nucleus. So by the time we get back around to where this number 6 is, we have 2 daughter cells. They have the exact same cellular components. And then each of those daughter cells goes through this cell cycle and divides and makes daughter cells that go through the cell cycle and divide. So this big piece that goes all the way around from number 1 to number 4 is interphase. An interphase contains gap 1, S, and gap 2. Then from number 4 around to number 6 is mitosis, and mitosis contains prophase, metaphase, anaphase, and telophase. And really this comes about because there were people that were studying cell division. So they were studying this piece. They were studying this mitosis piece. This is the piece that you can see under a microscope. You'll see in just a second. And so we had mitosis people, and they were studying mitosis, and they were putting it into stages. They were looking at cells under microscopes, seeing what stage of cell division the cell was in, classifying and trying to do all this research and finding out all of the events that occur in mitosis. And from their point of view, looking under a microscope, this whole other time didn't look like the cell was doing anything. Looked like the cell was just sitting there. That's why they called it interphase. It's in between the cell divisions. They didn't see anything that the cell was doing under the microscope. Then you have some biochemists, and the biochemists are studying what happens when the cell is making proteins. Transcription and translation. What happens when the cell is making DNA synthesis or replication? What happens when the cell is getting ready to divide? And so they had a separate set of terminology for their biochemical steps, for the protein synthesis and DNA synthesis steps that the cell was going through. And they were doing biochemical experiments rather than looking at the cell under the microscope. So that's how we end up with this kind of two different sets of terminology, G1S and G2, and then prophase, metaphase, anaphase, and telophase of mitosis. This is a good figure to see how those fit together, and I'm hoping that that starts to eliminate some of the confusion of what we're talking about. So if we divide the cell cycle up, we can divide the cell cycle into interphase, which is where it doesn't look like anything's happening under the microscope, but we're doing a lot of growing, making proteins, making DNA. Then the phases of mitosis, and then cytoplasmic division. Or if you're just looking at mitosis, then you're looking at these prophase, metaphase, anaphase, telophase steps. And that IPMAT, if you can remember that, that helps you put them all in order. Interphase, and then those steps of mitosis. Interphase. Interphase, under the microscope, it doesn't look like anything's happening because the proteins in the DNA are submicroscopic. You can't see them with a light microscope. So they just thought this was an interval between cell divisions. They just thought the cell was sitting there not doing much, or maybe it was growing and it had to be replicating its DNA. So this interphase is the phase where the cell makes new organelles, makes more cytoplasm, and replicates its DNA. Now we know that the G1, or the first gap phase, is where the cell is growing. S phase is where DNA is replicating. And G2 is where the cell is preparing to divide. Then we can look at the steps of mitosis. Keep in mind that mitosis is a continuous process inside the cell. We're dividing it into stages in order to describe these major events, but it's really a continuous process of division. I've got several animations that are in the animations tab in the module folder that I think that you might think are helpful. One of them is the same as the animation that's in the PowerPoint that you can't get to play. I've not figured out why that didn't copy in the way that I had wanted it to, but that video, the same video, is available on YouTube at this link, or it's available at in the animations tab so that you can take a look at that. So this is the figure in your book that's showing what's happening. The first thing that happens is that the chromosomes condense. When you look at this under a microscope, this is the first time you can really start to see individual chromosomes. The nucleus starts to look like gummy worms. In interphase, the nucleus is just kind of a purple ball. You can't really see much about it under the microscope. That's why they didn't think there was much happening. It doesn't look like much at all under the microscope. And then in prophase, the chromosomes start to become individual units that you can see. They kind of look like gummy worms in the middle of the cell. So that's why the people that were using microscopes were interested in viewing this whole process. And in prophase, the chromosomes become visible in the beginning, in the center of the cell. That nuclear envelope starts to break down, and so these chromosomes are just kind of floating in the inside of the cytoplasm. So the next phase that we're interested in is metaphase, and the hallmark of metaphase is that the chromosomes are lined up in the center of the cell along a line that's often called the metaphase plate. So when I was remembering this process and trying to remember the steps in order, for some reason that metaphase plate term really stuck in my head, so I could get metaphase, and then I could work kind of forward and backward from metaphase. So whichever step kind of makes the most sense for you, use that kind of as an anchor, and then figure out the other steps around whichever step makes the most sense. And for me, it was the metaphase. I could see when I had to look at this in a lab, I could see the cells lined up in the center of the cell, and so that made sense to me. Now, another organelle that we just talked briefly about when we were learning about organelles was the centriole. The centriole plays a pivotal role here in mitosis. You'll see, if you look at that YouTube video, you'll see that the centrioles have proteins called microtubules, and the microtubules attach to each of the sister chromatids of the replicated chromosomes. And those microtubules are going to pull those chromosomes apart in anaphase so that one of those chromatids goes to one cell and the other chromatid goes to the other cell. And then telophase reforms the nuclear envelope around each of those new nuclei. And then the last part of the cell cycle is going to be cytoplasmic division. So if we go back to here and try to put some of the events that we've talked about with what we see here, in the G1 phase, we'll have this one chromosome. In the S phase, we'll replicate this chromosome. A microtubule will come along. A microtubule will come along and attach itself from a centriole to each of these chromatids and pull them apart to the daughter cells. The microtubules attach to the centromere in metaphase to line the cells up along the metaphase plate, and then the pulling apart occurs in anaphase. So that kind of links what we talked about with the chromosomes to what we're talking about in each of the steps. Cytoplasmic division is different in animal cells than plant cells because of the difference in structure between animal cells and plant cells. That was some of the material that was in Chapter 3. One of the big differences is that in plant cells there's a cell wall. Cytoplasmic division in animal cells is to be an outside-in process. A cleavage furrow forms and then the two cells kind of pinch apart. In the plant cells, it tends to be a more inside-out process. There's vesicles that contain the components of the cell wall, and those vesicles tend to fuse inside the cell to create a kind of a cross wall between the two nuclei. So I give you a set of links to some time-lapse videos. I don't have all of those up in the animations tab yet, so that mitosis in real-time slide, you might need to go and cut and paste those into YouTube, but it'll take you through and actually show you under the microscope what that looks like. And then that is up in the animations tab to the mitosis video. That's really helpful to try to put all of those steps in order that we just looked at. So now I want to go through some of the things related to genetics. And one of the things that you'll find as we go through this part is a lot of the terms occur in pairs. There's a lot of the terms that you'll find two terms that occur in pairs, and that makes it somewhat difficult to tell the difference between the two terms. For example, what's the difference between a gene and alleles of a gene? You'll see gene and you'll see alleles. They're not really interchangeable, but you'll kind of see them see some of the terms in pairs like this. So I'm going to try to go through some of these pairs and kind of explain a little bit what they mean. So our homologous chromosomes are the ones that were the same. You got one from a dad and one from a mom, and one from a sperm, one from an egg, one from a female parent, one from a male parent. And they're very, very similar in their structure. The only difference might be a slight variation in the DNA sequence at the different locus where a gene is. A gene codes for a specific protein that might influence a specific trait. An allele is just a different form of the gene that encodes slightly different versions of the gene product. Okay, so what does that mean? So I ask you, so what are gene products? Well, when we talked about gene products, we were talking about things like making a protein or even making an RNA that becomes a protein. But most of the time when we're talking about gene products, when we talk about gene products, we're talking about proteins. So if you have a gene for something like eye color is produced by a protein called melanin. So the gene for eye color makes a protein called melanin. You have an allele for blue eyes. You have an allele for brown eyes. You have an allele for green eyes. So those are slightly different forms of the gene for melanin. So the alleles are the variations in those forms for the gene for a trait. Now that's kind of simplified because they have found in humans that there are a couple of genes that affect eye color. There's a gene for melanin, and then there's a gene that determines how much melanin gets made. So it's not quite as simple as I've represented it here for eye color, but I'm using that as an example to try to help you understand the difference between gene and allele. So you may have a gene for the protein in hair, keratin, and you have an allele that codes for a structure of keratin and collagen that forms curly hair, and an allele for the structure that forms straight hair. And so for that gene, you have different versions, and those versions are called alleles. And alleles are the basis of the physical traits that we see. Now, another set of terms that you may see is the term haploid and diploid. Haploid just means you have one of each type of chromosome, and diploid means you have two of each type of chromosome. We just talked a little bit about mitosis, and your book also talks about meiosis because it divides the chromosome number in half. It takes a diploid cell and it makes a haploid cell out of it. To help with that idea a little bit, I have this figure. This is a figure from a different textbook. This is also in that other PowerPoint. And so we use n here as a variable, like x or y or whatever. N just means the number of chromosomes that are in a sex cell. Whatever the gametes are for that species that you're looking at. So our body cells are diploid. Our body cells have 46 chromosomes. They have a pair of each chromosome. So there are 2N. And so we grow from a fetus into an adult, still 2N. Then we undergo meiosis. If we're female, we make an egg cell. If we're male, we make a sperm cell. But the process of meiosis divides in half the number of chromosomes. So these gametes are haploid. Where our body cells were diploid, our gametes are haploid. So we put a sperm cell and an egg cell together, and that forms a zygote, and that zygote now has the appropriate number of cells. And then I made a joke about singing Circle of Life. This has to happen for the same reason that we have to replicate chromosomes. But here we're having to divide the number of chromosomes in half, because if we didn't, if we took a 2N cell and we put it with another 2N cell to make offspring, that offspring would have 4 times the number of chromosomes. And if we put 2 of those together, they'd have 8 times the number of chromosomes. And if we put 2 of those together, they'd have 16 times the number of chromosomes. So pretty soon, by merging gametes to make offspring, we'd end up with too many chromosomes. When we divide cells, we would end up with too few chromosomes. Here you'd have the opposite problem. So meiosis halves or divides in half the number of chromosomes so that we can have this cycle where we have an N male gamete and an N female gamete come together to make a 2N that grows up to an adult that makes an N gamete from a female and an N gamete from a male, and they can come together and produce a zygote. And that's the life cycle of humans. And so meiosis takes a diploid cell and makes it into 4 haploid cells so that we can put 2 of them together in fertilization and end up with a diploid cell. Now, a lot of the mechanisms of meiosis are similar to those of mitosis. So I wouldn't get too bogged down in a lot of the details of meiosis. I would try to more understand overall what's happening using the previous figure that I showed you and this slide that's helping you to see kind of what overall is happening. In meiosis, we're dividing in half the number of chromosomes in the cell. Again, there's a video in your PowerPoint that if you click play, it tells you it can't find the file. So if you'd like to see that same video, I give you the link here, and it's also linked in the animations tab in the module, so you would be able to find it there. And so talking about meiosis, talking about putting an egg and a sperm together to make a zygote, and talking about some things like crossing over or some of the ways that we get genetic variability can lead us into a discussion about inheritance of the genes that are on the chromosomes in that offspring, in that zygote. And there was a person that worked on this. His name was Gregor Mendel, so that's why we tend to call this Mendelian genetics. That's not actually how you spell Mendelian, but that's okay. He was a monk, so he lived in an abbey, and he really didn't have much to do except for play with pea plants, and so he played with pea plants. What he did was he used what he called true breeding strains, or plants that gave the same color flowers generation after generation after generation. And pea plants were very easy because they don't self-fertilize. He actually had to rub the parts of the plants together to get them to fertilize. They grow really quickly, so you can see what the results of your crosses are. And you could manipulate that to see what your outcomes were. And so he used strains that gave the same offspring generation after generation in his experiments. We now use the term homozygous to describe the chromosomes and the genetics behind what he was using as true breeding strains. So that leads me to a couple more pairs of terms. One pair is phenotype versus genotype. Phenotype is the traits that you can see, the physical traits. Phenotype is the physical traits. When a gene gets expressed, we see that in the traits. When a gene actually becomes a protein, we see what the person's eye color is. When a gene actually becomes a protein, we see how curly the person's hair is. So the gene expression results in the phenotype, physically what you see. And then the genotype is what genes or what alleles of the genes were there. So that brings us back to our diploid term. Diploid cells have homologous chromosomes, usually one from each parent, and they have pairs of genes. So you have a gene for hair color from your mom and a gene for hair color from your dad. What specific alleles are there is your genotype. And then what you look like is your phenotype. The alleles that are there make up the genotype. You could have the same two alleles. So we could say you're homozygous for the gene. Or you could have two different alleles and we say you're heterozygous. So that brings us to this pair of terms, homozygous versus heterozygous. If you have the same two alleles for that gene, you're homozygous. For example, if you get the allele for blue eyes from your mom and the allele for blue eyes from your dad, you have the same alleles for the gene for eye color. You're homozygous for the gene for eye color. If you have two different alleles, you get the allele for brown eyes from your mom and the allele for blue eyes from your dad, we say you're heterozygous. Homo means same, hetero means different. So if you have the same two, you're homozygous. If you have different two, you're heterozygous. And so this shows you two homologous chromosomes. It shows you that the same genes are on each chromosome. They could be the same or they could be different. If they're different alleles, they're heterozygous. If they're the same alleles, they're homozygous. And then another set of terms that tends to come in here is the term dominant and recessive. We can describe an allele by saying it's dominant or recessive. What determines if an allele is dominant or recessive, we can look at an individual that has both alleles. We can look at the heterozygous individual and see what their phenotype is, see which allele is expressed. So if you have the allele for brown eyes and if you have the allele for blue eyes, what color are your eyes? That tells us which allele is dominant to the other one. The terminology can get very confusing. And so the concepts don't make sense until the terminology makes sense. And so whatever, if you want to make just a list of the terms in pairs, try to put in your own words what the definition of the term is, and that'll help you. Or if you want to make note cards, or if you want to use sticky notes, or whatever works. But understanding the difference between the terms in a pair and the terms in your list is going to help you to learn the concepts, because the terms help convey the concepts. So however you can distinguish between the gene and the allele of the gene, however you can distinguish between genotype and phenotype, homozygous and heterozygous, and dominant and recessive, those are going to help as we go through and talk about some of the other concepts that we're going to talk about. So Mendel worked with pea plants, and he was looking at a number of different characteristics. He looked at seed shape, seed size, whether or not the seeds were smooth or wrinkled, the pod shape, the pod size. He looked at how tall or short the plant was, how long or branching the branches were, and he also looked at flower color. And the plants that he used either made purple plants or white plants. And so this is the example that your book gives for one of the crosses that he made. So if we're going to look at our terms, the gene that we're talking about here is the gene for flower color. And there are two alleles. One makes purple flowers. The other makes white flowers. So those are the two different versions of the gene for flower color. We're using the letter A to denote these alleles. We tend to use a capital letter to denote the dominant allele, and we tend to use a lowercase letter to denote the recessive allele. So the dominant allele we will denote with a capital A, and the recessive allele we will denote with a lowercase a. And we can figure out which is dominant and which is recessive by looking at the heterozygote. It has both alleles. It has the allele for purple and it has the allele for white. Whichever one got expressed, whichever one is seen as the phenotype, is the one that we say is dominant. So if the AA flower is purple, that tells us purple is dominant. So we can go back up here and match phenotype with genotype. So the dominant allele codes for purple flowers, and the recessive allele codes for white flowers. Now, a Punnett square is usually used to predict what offspring can be made from. So if you know the genotype of your parents, you can predict the genotypes of the offspring. This takes us back a little bit to meiosis and going from a diploid to a haploid cell. So if the person's genotype has a dominant allele and a recessive allele, has a big A and a little a. When they make gametes, some of those gametes will have a big A allele, and some of those gametes will have a little a allele. And so that's what they're showing you here. They're showing you some of these gametes will have a little a, and some of these gametes will have a big A. Because if you take those two chromosomes with those two alleles and you separate them into gametes, one gamete will have one, and the other gamete will have the other. Same with this other parent. Doesn't matter which parent goes on top and which parent goes to the side. If you're talking about humans and you're talking about males and females, it doesn't matter. Just one parent goes to the side and one parent goes to the top. And then over in Part A, they actually show you how to fill in the Punnett square. So the square that is the combination of these two alleles goes here. The square that is the combination of these two alleles go here, etc., etc. And that's how we fill in what the offspring are. So, if this is one plant's egg cell and this is the other plant's sperm cell, then the offspring will be big A, big A. So they show you in Part A how to fill in the Punnett square, and then they show you in Part B what the Punnett square looks like, and they show you both the genotypes and the phenotypes. The phenotype is they show you what color the plant is. They show you a picture of the purple flower or the white flower. And then the genotype is the combination of the two alleles that this plant got from fertilization of the gamete from one plant and the gamete from another plant. Now, a lot of where this becomes helpful to us is not when we're talking about plants. This tends to become helpful for us not when we're talking about pea plants or what Gregor Mendel did in a abbey as a monk a number of years ago, but when we're talking about human genetic disorders. Figure 916 is showing you an example of a disorder that's carried by the dominant allele. And so they're showing you in red the allele that causes the disease. So the father has the disease. He has the diseased allele. For a dominant disorder, just having one copy of the allele is enough to get the disease because that allele is dominant to the other one. It doesn't matter what the other allele is. That allele will be expressed. That allele will be seen as the phenotype. So this is similar to that purple allele, that dominant allele in that previous slide. And so we can take this father and put him along the top of this Punnett square. We can take the mother and put her along the side of the Punnett square. And we can determine what the offspring are. Now, to tie in a little bit of our terminology, the father is heterozygous. He has two different alleles. He has a diseased allele and a normal allele. The mother is homozygous. She's actually homozygous recessive. She has two recessive alleles. She has two lowercase letter alleles. So she's homozygous recessive. So when she makes eggs, she's only going to make one kind of egg. It's going to have a little A in it. It's either going to be this little A or it's going to be this little A. Either way, she only makes gametes that have a little A. The father can make 50% of his gametes with the diseased allele and 50% of his gametes with the normal allele because he's heterozygous. He has one of each. And then if we match up these two alleles, then we get this genotype. If we match up these two alleles, we get this genotype. We match up to get the other ones. I don't want to draw all over it because then you won't be able to see. Hang on, Robin. I'll get to your question in 1 second. So the outcome of this is that 50% of the children are going to have the dominant allele and 50% of the children are not going to have the dominant allele. And what they've shown you with the green background are the children that will inherit the disease. But they show you with the white background are the children that will not inherit the disease. Now, this is actually just showing you, because of independent assortment, because every time you get fertilization, you get a random sampling of eggs and a random sampling of sperm, and that's a random sampling of the chromosomes that divided. Really, this is just telling you for each child, there's a 50-50 chance that that child will have the disease. So this is not telling you if you have 4 children, 2 of your children will have the disease and 2 of your children will not. It tells you every time you have a child, there's a 50-50 chance that child could get the disease. So you could have a child that's normal, genotypically and phenotypically normal, and that doesn't guarantee your next child is going to have the disease. You could end up with 4 kids that are genotypically normal. You could end up with 4 kids that inherit the dominant diseased allele. Sorry, I had to cough. So, yes, Robin, to answer your second question, that is why a couple that has several children, they could have all children that get the disease. They could have all children that do not get the disease. If they are concerned, and if there's a strong possibility for inheritance, that's one reason why they do amniocentesis. Amniocentesis is where they can genetically sample the child while it's a fetus, still in utero. If the parents think that they may want to abort the fetus based on the results of that, that's why some people do amniocentesis. Other people just do it because they want to know, they want to be prepared for the genetic outcome of the child. They wouldn't make those decisions, but they just want to know. They can also do that genetic testing at birth. Some of these dominant alleles we test for at birth. Some of them, well, you can test the mother and you can test the father, but all you'll know is this here. You won't know which specific sperm is going to meet up with which specific egg to give until that fertilization happens. So the earliest you could know about that specific child would be amniocentesis in utero. Danielle, right now we are talking about a dominant allele, so anyone with that dominant allele will express that phenotype. In just a second, we're going to talk about the recessive, where that's not the case. So you are correct. In this case, because it's a dominant allele, the father will have it, show it in his phenotype, and the offspring will show it in their phenotype because this is a dominant. So let me show you the one for the recessive. So figure 917 is for the recessive. This is a disease that's inherited by a recessive allele. allele is normal and the recessive allele is diseased. So let me type that up so we can keep that in mind. In this case, because the dominant allele is normal, and if you have at least one dominant allele around, you'll have that dominant phenotype. Here, with a recessive disease, you have to have both copies of the recessive allele to have the disease phenotype, which I just abbreviated PH because it would run off the side. So that's what makes this a little bit different from the dominant. The dominant, if you just had one allele, you had the disease. Here, you have to have two recessive alleles in order to have the disease phenotype. So to answer Robin's question, yes, and that's why they show you, that's why they use this term carrier. Both of these people, both of these parents, are phenotypically normal. By their phenotype, they do not have the disease. They may not know they're a carrier. They may not know they carry one normal allele and one disease allele. And so these two people would make these sets of gametes. The father could have a dominant allele in some of his gametes and a recessive allele in some of his gametes. Mother would be the same way. And so there's a chance that the child will get two dominant alleles. The child would have two normal alleles. They would not even be a carrier for the disease. There's a 25% chance that this child would have that genotype. There's a 50% chance the child will get one dominant allele that will allow them to have a normal phenotype, and they'll get one recessive allele, which makes them a carrier for the disease, but they would not have the disease themselves. They wouldn't phenotypically have the disease. They would just carry the gene for the disease. And there's a 25% chance that the child would get both recessive alleles, and in that case, they would exhibit the disease phenotype. There are several diseases that are inherited in this autosomal recessive fashion. Some of them you might be familiar with. We've talked quite a bit about cystic fibrosis. I've talked to some students individually, as well as we've talked about as an example and in some of the answers that we've done. So there's a lot of autosomal recessive disorders. I think I have. Yeah. So there's a table in your book. This is table 9.2 that shows you some of the disorders that are inherited in this autosomal recessive. Galactosemia and phenylketonuria, we tend to test for at birth now because they can cause pretty significant impairment. Cystic fibrosis is one that's inherited in this autosomal recessive fashion. Albinism is inherited in autosomal recessive fashion. So this autosomal recessive is how you can have two parents that look normal and you have offspring that end up with the disease. Because there's this 25% chance that the offspring will get both of the recessive alleles. Now we can look at what happens if this carrier parent, we can cross that carrier parent with a person that is not a carrier, and we can do that kind of a Punnett square. So we would still have the same alleles at the top, but then we have the carrier, the parent who is not a carrier does not have any diseased alleles. And so if we look at what the outcome of that is, There's a 50% chance that this child would not even get a diseased allele. They would be both phenotypically normal and have two dominant alleles. And there's a 50% chance that they would be a carrier. They would be physically normal. They would phenotypically normal. But they would have the allele in their genotype. They would be a carrier for the allele. So it's really when both parents are a carrier that you can end up with this affected child, the child with both recessive alleles. And so that's one of the differences between the recessive inherited traits and dominant inherited traits. In a dominant inherited trait, you have the disease if you have just one allele. In the recessive inherited trait, you have to end up with both alleles in order to exhibit phenotypically the disease. And the last kind that we can talk about are sex-linked traits. We didn't talk earlier, but the way that sex is determined is that last set of chromosomes, that 23rd pair of chromosomes that are not homologous chromosomes. They're not homologous because you can either be XX or XY, and if you're XY, those two chromosomes are not homologous. So females are XX and males are XY. So when females go through meiosis and females make eggs, females can only contribute an X chromosome to those eggs. It's either this X or it's this X, but either way, all eggs are going to have an X chromosome in them. For males, when they go to make gametes, they can either have this X in their sperm or they can have a Y in their sperm. So males actually determine the sex of the offspring. If you have an X sperm that fertilizes an egg, you get an XX, which is a female. If you have a Y sperm that fertilizes an egg, you get XY, which is a male. So it's actually sperm that determine the sex of the offspring. So your book talks about some X-linked recessive disorders, which are recessive diseases that are found on the X chromosome. Your book makes the statement alleles on the X chromosome are inherited differently in males and females, and that's what I was trying to show you down here. Females will get two X chromosomes. Males will only ever have one. So if it's a recessive disorder, females would have to have two recessive alleles on their X chromosomes. Males would only need to have one because they only have one X chromosome. That gene is not going to be on the Y chromosome. The Y chromosome is different. So females pass those X-linked alleles to their male offspring to a much higher degree than they pass those X-linked alleles to female offspring. Both the mother and the father would have to have the X-linked recessive disorder to pass it to female offspring. Females pass the X-linked trait to male offspring in a higher rate. And so here's a table that gives some examples. Red-green color blindness is pretty common. Some of the muscular dystrophies. Your book goes through a story of the Queen Victoria of England and the family that all the boys have hemophilia. That's a pretty classic example of what we're talking about. So here, instead of using A's to denote the alleles, we have to use X and Y to tell you which chromosome we're talking about. The gene is only going to be on the X chromosome. The gene is not on the Y chromosome. So here they're denoting, again, the disease allele in red. So we have a mother who's a carrier. She could be phenotypically normal. She makes gametes. Half of them have this X, half of them have this X. So half of them have the disease allele on the X chromosome. Half of them have a normal allele on the X chromosome. And then mother makes gametes. 50% of them have a Y chromosome and 50% of them have an X chromosome. So when we match them up, half of her daughters may get or there's a 50% chance that a daughter would get the disease allele, but there's a normal dominant allele from the father. Since this is a recessive trait, you'd have to have 2 copies, and so it's unlikely that the females are going to exhibit the disease phenotype. There's a 50% chance her daughter will not be a carrier. There's a 50% chance that the mother will pass on the normal allele to the son. There's a 50% chance the mother will pass on the disease allele to a son. And in this case, the son would be affected with the disease, hemophilia or color blindness, or some of those types of traits that we're talking about. So those are some of the examples that I wanted to go through. It's interesting because you hear a lot about inheritance of diseases. You hear a lot about where they come from and sex-linked traits and inherited traits and people being a carrier. And it's when you actually get into studying it that some of those things that you hear about make a little bit more sense. So I'm glad that you guys asked questions. I hope that I answered all the questions that you had. If you're looking more into this or you're studying more into this and you have questions, go ahead and post them on the discussion board, because there's a lot of times that people will think of scenarios or think of questions about inheritance. In my Bio 130 class, we're going through and talking about inheritance of blood type, and they've had a lot of questions about inheritance of blood type. Can somebody with this blood type have offspring that has this blood type, and those kinds of questions that we've had come up. So it's been a really interesting discussion about inheritance from the position of inheritance of blood type. So Danielle is asking about eye color. If both parents have brown eyes and their son gets green eyes, that means that they were both carriers. That's correct. That's a good way to put it. We tend to use the term carrier more for things like diseases, but the concept of what you're saying is absolutely correct. Both parents were heterozygous. Both parents had a dominant allele and a recessive allele, and those 2 recessive alleles came together in order to give the eye color. So it's going to be similar to, I clicked the wrong one, hang on. It's going to be similar to what we talked about with a recessive disease, only instead of disease, you're talking about eye color. Green eyes is not a disease. It's not going to kill you. So probably if 2 parents with brown eyes had a child with green eyes, if we're talking about simple inheritance patterns, then both parents were heterozygous, and then the child inherited the recessive alleles, and that's how they ended up with green eyes. Now, they have come out with the idea of epistasis for human eye color, which means that it seems like more than one gene is involved in human eye color. So it's not going to be quite that simple. You have 2 parents with blue eyes that has a child with brown eyes, and you wonder where that dominant allele came from, and there's actually several different genes that can be involved in that process. But if we're looking at it in the simple inheritance terms like we're trying to learn it right now, what Danielle says is correct, and it's based on this model right here. If brown is dominant, then the fact that they have the dominant allele makes them have brown eyes, since the child does not have a dominant allele, then they express the recessive trait, the green eyes. You are welcome. So what other questions do you guys have? Some of the other things that your book talks about aneuploidy, which is where you're missing some chromosomes. Talks about polyploidy, where you end up having some extra chromosomes. Those are pretty straightforward. There are some environmental effects on phenotype. And we're going to start getting into that in the next couple of chapters when we talk about natural selection, because in order for an organism to adapt, they have to have a genetic basis. You don't just, I need a gene to blend into the sand, so I just automatically make a gene that blends into the sand. But if there is a gene in the population for blending into the sand, then that will start to be expressed in higher and higher levels. And so the examples that your book gives are about the plants that are grown at different elevations and the Himalayan rabbit, the coat color in the Himalayan rabbit. That's kind of an interesting one too. So that's kind of a preview of what we're going to talk about next week. So Robin is asking about genetic testing. You can, at this point in time, I think you have to pay for it. I don't know that most insurances pay for it. You can get your entire genome sequenced, and that will tell you if you have disease alleles for the diseases that we know that can be attributed to genetics. Unfortunately, it's not quite that simple. There are people that have, for example, the BRCA genes that they found that are linked to breast cancer. And some people have those genes and have not yet developed breast cancer. Some people do not have those genes and develop breast cancer. There are a lot of other things that are involved in some of the diseases that we're talking about, especially with cancer. There's a lot of environmental components of cancer. So drinking high fructose corn syrup seems to be as big a risk factor as having the BRCA gene. So if you go to McDonald's and drink a Coke a lot, or if you're eating food with a lot of high fructose corn syrup, then you might be at a higher risk for somebody that has the BRCA gene. And there's a lot of times that they recommend that like Danielle is saying, you have to get tested to at least know. And knowing that you have a gene can help you try to minimize those environmental factors. But there are a lot of genes there's a lot of diseases that it's an interplay between environment and genetics. And knowing that you have the gene does not mean that you'll get the disease, especially if it's a progressive kind of disease. Now, you can find out things like the autosomal dominant diseases. Having just one copy of the allele tends to lead to that. So if you have a copy of that allele, you can be pretty confident that you'll pass that along to your child. But again, my mom is homozygous dominant for the hypercholesterolemia gene. So this familial hypercholesterolemia, high cholesterol level in the blood. So my mom is homozygous dominant for that. So she gave me one of those. I know I've got at least one of those genes. I don't know what I got from my dad, but I know I got at least one of those. So that tells me that I need to do quite a bit of diet and exercise and environmental controlling to try to prevent having a lot of high cholesterol in my body. But having that gene does not necessarily mean that that will contribute to me having the disease. The other issue is, as we start finding out more and more about our genes and what our predispositions are, insurance companies can start to use some of that information to try to exclude people. And that's part of what the Health Care Act wants to address, and there are protections that people are trying to put into place. So you can get information about what genes you would pass on to your offspring, but then what happens if you can't get insurance or your offspring can't get insurance because of what genes your insurance company knows that you have? And controlling that information is going to be key. So I personally have not chosen to do genome sequencing because I'm not confident enough in the controls that they kind of have out there. What happens to people that are adopted with a sealed adoption record? I'm not sure about a lot of that information, and that's one of couples that try to reproduce using anonymous sperm donation and anonymous egg donation are automatically at a disadvantage about knowing a lot of that medical information. Sealed adoption is going to be very similar. I don't know the legalities of sealed adoption. I don't know, but I do know that there are anonymous sperm banks that will not that will not release medical information, and there are some that won't release till the child turns 18, no matter what kind of health issues they have. So, Marvin, did you raised your hand. Did you want to have some input?
Yes, all three of my children were adopted, and from 3 different states, and it's pretty common that each state tells any hereditary type diseases with the child. That is pretty much automatic with each state.
Okay, that's good to know. I have more experience with sperm and egg donation than I have with adoption. I know there's a lot of sperm banks that will give any known hereditary information from donors that they have. Even if it's anonymous donation, they'll at least tell you, there's a family history of breast cancer, there's a family history of heart attack. And my understanding is in the United States, there is more of that information that might be available versus international adoption. I don't know how much of that information, I don't know much about that. I'm kind of talking more outside of my experience. Marvin has more experience than I do, apparently. That's good to know. I think that having the medical information is really important for both for yourself and for offspring. You always hope that you don't need that information, and you always hope that you don't ever have to make decisions. But I think that it's important to children to have the information that they need. What else? Other questions that you guys have? Yes, thank you, Marvin. That was helpful for the discussion because that's, like I said, that's kind of outside my realm of experience. So that's good to know and have that input. That's a good discussion, you guys. This is usually one of the topics that people have experience with, and so this is usually one of the topics that people want to know more about. So this usually elicits some discussion. So that's good. That's not where I wanted to go. I wanted to go back up here. Okay, well, I will go ahead and make sure the archive is available. And if there's any other questions or anything that come up, let me know. And if there's any questions that come up about the assignment is just kind of a series of questions this week. So hopefully that'll be pretty straightforward. If there's any questions that come up about that, be sure and post them. So thank you guys. It was a good discussion.